It's been 24 hours since since I set up the experiment. I just uploaded the video of setting up the experiment like 20 minutes ago. So if you haven't seen that before, check that out. What I have here is three samples. One, two, three. I have the indole acetic acid sample, the naphthalene acetic acid, and the control. And what I'm going to do is I'm going to do a hemocytometer count of each one of them, and I'm going to record that count of each, and then I'm going to take some more sample and I'm going to put it on these three slides, and then I'm going to actually do some measurements and some observations of the changes to the algae. Now, right off the bat, there's definitely some observable changes. The biggest change is with the naphthalene acetic acid. When you just looking at the culture from with no grow light, so just regular office lights, it looks pale compared to the indole acetic acid, or compared to the control and the naphthalene. Now, one thing that's interesting is that under the grow light, which has blue and essentially blue and red lights, essentially, the naphthalene acetic acid looks dark, which would indicate that it's actually absorbing a lot more light. Now, you don't see it under the white light because chlorophyll only absorbs certain wavelengths, but with the grow light, those are the wavelengths that it absorbs. I have never seen that before. It looked like it looked like somebody put the algae through a blender. And this is only 24 hours. I don't know if it was... I'm gonna have to do some but anyways, I can't even make a count on this. So what I'll do is I'll take that one, clean it out, and then redo it and do the indole or the naphthalene acetic acid count. So now we're going to do the naphthalene acetic acid. I'm pretty sure I'm saying that right. Naphthalene. It's a tongue twister. It's like PHTH. But let's see if I can. Oh, okay. I see something here. 1, 2, 3. Now, right off the bat, I don't see any changes in structure. So, okay, I got a count of 21, which, let's see here. 21 times 10,000. Okay, 13 million. I even see some new babies. Those are those little ones. And I got 31. That's interesting. Now let's do the math. Just over 19 million. It seems like the hormones aren't actually that great for the algae, but that's preliminary. It's only been 24 hours. Now we're going to look at the indole acetic. Now this is where the my program is really cool here. Not my program, I didn't design it. the AmScope program, because I can actually take pictures if I have something to take pictures of. There we go. And what I'm doing is I'm taking some pictures. And let's see here. Let's go to 400. It looked like they just blew up. Now let's see if I can do a... What is that? Because this right here looks like it seems like it's a somewhat intact algae, but it just looks like the cells just essentially here's another one. But let's do the let's see, 100, 4,000. Let's try the 4,000. Okay, so that's enough of that. Let's move on to the other ones. Now these look pretty unremarkable. And then I'll do the control. 18. Now I'm trying to find bunches of algae, that way I can take measurements of a bunch at a time. Now I'm not going to bore you with measuring every single one of them, because for example, I actually just uploaded a data set up on to Mendeley, one of those, with over 1,100 measurements for trickle measurements, and as a baseline. So it can get pretty tedious, but I'm going to show you essentially how it works. There's this little tool where I can click on one side and then pull it, and this one's 0.1 millimeters. And what's really cool is if I do this on an entire picture, I can actually take those measurements and download them to a spreadsheet. So once I do the measurement on one picture, it appends it to that spreadsheet, and I go to the next picture and I keep going, and I have a whole database of measurements that I can then play around with, however I need to. But rather than having to store all these, I can just make those measurements and then put them into a spreadsheet and then figure out what to do with them from there. Well, that's pretty much it for this video. We'll see you. It'll be, unless I do something in between, it's going to be another 3 days before I take another observation and add some more hormones. And I'm going to keep the indole acetic acid thing going as far as until I realize that until I can really tell whether the culture has completely collapsed and died. For right now, I don't know if maybe there's just those kind of cells can somehow there's enough cells there to maybe regenerate. But who knows? We'll check it out. Make sure to subscribe and do all that cool stuff. Thank you.